Today we shall discuss about the effect of immobilization on functional properties of the enzyme molecules. We have seen a number of immobilization methods that we have discussed in the previous classes. We see that in most cases the enzyme undergoes a loss of activity during immobilization. In fact, no method can ensure that you can recover the enzyme in 100% yield. The yield can vary depending on the method of immobilization, depending on the carrier and the stability of the enzyme itself with respect to the conditions of immobilization. Now, this loss of enzyme activity could be real or could be apparent. By real means, by real I mean that you cannot recover after the immobilization the original enzyme activity by physical interventions. On the other hand, the apparent loss refers to that apparently you are measuring a reduced enzyme activity, but by certain physical interventions the original activity can be restored, and essentially it is not a permanent loss of activity, but due to certain physical conditions, to physical forces which make an interplay with the catalytic function, the enzyme activity seems to be apparently appearing to have been lost. And so now the reasons for the two types of losses, real and apparent, could be attributed to one or more of the following four factors that are listed here. And first and foremost, the factor is the change in the enzyme conformation itself. As you will notice that when the enzyme, a soluble enzyme, is immobilized onto a matrix, the interaction between the matrix and the enzyme amino acids might lead to certain conformational changes, possibly due to the result of involvement of some of the amino acid residues in the mobilization process. So therefore, all the intermolecular forces that attribute to the enzyme conformation are altered, and so ultimately very often it has been noted that a very common feature is that upon immobilization an enzyme may undergo some kind of unfolding, some kind of a stretching. Of course, there are no quantitatively measurable sort of parameters which can be simply measured. Of course, a detailed analysis or detailed study on the conformation, just like the what we do as far as identification of the globular structure, the tertiary structure of the enzyme, I mean one can study, but otherwise a quick quantitative measure of the conformational loss is only through measurement of enzyme activity. And so for example, here you see that this is a typical soluble enzyme shown here. It has been immobilized onto a solid matrix by some bonds. It could be by electrostatic bond, it could be by metal salt linkage, or it could be covalent bond, or it could be also as an entrapment within a gel. Now, as I mentioned earlier, that entrapment within a gel fortunately does not involve the amino acids of the enzyme protein, and therefore the chances of any conformational change during gel entrapment are minimum. Then during immobilization the enzyme may undergo some conformational change. Just a hypothetical change I have shown here. It could be I mean in any shape, it could be simply unfolding, it could be some kind of a stretching, and essentially it may lead to a totally inactive enzyme preparation or a reduced activity of enzyme preparation. By reduced activity I mean that while the protein content will be reduced, the specific activity of the enzyme, that means the enzyme activity in terms of international units per milligram of protein, might get reduced. Of course, there have been certain exceptions. I am saying exceptions, which are rare examples where the enzyme activity after the immobilization has also been enhanced. Again, if the conformational changes are more favorable to the factors that are responsible for reduction in activation energy, either by your strain or any other mechanism that we discussed earlier, by entropic or enthalpic changes which reduce the energy of activation for the enzyme catalyzed reaction, it might also lead to favorable conformational change, but those examples are indeed rare and not very common examples. Now then, the second factor which is responsible for loss of enzyme activity is steric hindrance. Now in many cases the immobilization process might involve the reduction in the access of the substrate molecule to the enzyme active site. You have achieved a good binding, but the enzyme active sites are hindered from access to the substrate molecule. Say for example, a typical case shown here. In the case the enzyme has been immobilized on sites which are in very great proximity to the active site, and therefore the participation of or the involvement of substrate with the active sites is reduced, and essentially that also results in reduced enzyme activity. However, such a enzyme activity can be recovered or can be this phenomena can be reduced if you can use a proper choice of the method for immobilization. As we mentioned that in some of the cases where the steric hindrances can be minimized by introduction of a spacer arm between the, for example, if you are going to immobilize this enzyme, you can introduce a kind of a hydrocarbon chain before it is really coupled to the enzyme molecule. That means between the binding sites, a spacer arm, introduction of a spacer arm which is usually a hydrocarbon chain and which can make the binding more flexible, and thereby the steric hindrances can be reduced. But in a particular method of immobilization, the steric hindrance results in a definite loss of enzyme activity, which is the real loss and cannot be recovered by physical methods. By physical methods.
In all cases.
Yeah, if the steric hindrance, for example, if you have covalently bound an enzyme by covalent bond, now depending on the method of binding, if you have not involved a spacer arm, the loss will remain there, but you cannot recover it. But of course, unless you modify your immobilization procedure, whereby a spacer arm is introduced, then you can reduce the steric. The method can be modified to result in reduced loss as a result of steric hindrance. But once a preparation has been made with a definite amount of steric hindrance, the loss is real. The third factor which contributes to loss of enzyme activity is essentially partitioning effect. Now, as a matter of fact, we must consider immobilized enzymes in the light of a shift from homogeneous catalysis to heterogeneous catalysis. Instead of now looking at the catalytic reaction in a homogeneous phase where enzyme is, the catalyst is dissolved in the same medium as the reactant. Here the catalyst has a different phase, solid phase, and the substrate is in the liquid phase. So basically you lead into a heterogeneous catalysis, and the various species that are involved in the reaction, it could be the substrate, it could be an inhibitor, it could be simple hydrogen ions which contribute to the pH. They might get partitioned in the two phases depending on the chemical nature of the bulk and the carrier particles, and this partitioning effect can also lead to an apparent loss of enzyme activity in a particular situation. Now if you look at, and the fourth is the diffusional limitations. Now a result of incorporation of a solid particle in a reaction mixture, and particularly we consider a porous particle, porous solid particle, where most of the enzyme has been immobilized on the interior of the particle, and of course when a solid particle is immersed into a liquid phase, there is also bound to be a stationary film of liquid around the particle. Now this film thickness can vary depending upon the physical properties of the substrate solution, for example viscosity or density. They may control the film thickness, and these might cause also diffusional constraints, particularly on this with respect to transport of the substrate from the bulk to the site of enzyme catalyzed reaction. Now the two effects, the partitioning effect and the mass transfer or the diffusion effect can be visualized here. If you notice here, for example, in the right hand side diagram, I have just assumed a hypothetical carrier preparation which is polyanionic in nature. That means it is highly negatively charged. An enzyme particle or enzyme molecule is entrapped or kind of located in the midst of negative charges. Now such a situation, and of course if the bulk, and consider situation in the bulk. Now the total charge inside the carrier particle or around enzyme where the catalysis is going to take place will be different than in the bulk. So obviously it will result to, and if in case if the substrate happens to be also a charged molecule, so there will be some partitioning effect of substrate between the bulk and the, so the real substrate concentration what we are considering in the bulk phase will not be same as that in the case of at the site of the enzyme reaction.
So
Yes.
what are the rod shaped things in the bulk?
No, rod shaped is only COO.
Sir, in the
The red.
the red ones.
In the no, I will come to that. I am just describing the right hand side one, and you see that there could be a very clear cut sort of partitioning effect with respect to hydrogen ions because they are positively charged, and a polyanionic carrier will definitely have a preference for hydrogen ions, and therefore the hydrogen ion concentration inside the carrier will be greater than the bulk. Similarly, if the substrate is also positively charged, you are going to have higher substrate concentration in the carrier or at the site of enzyme rather than the bulk. Or it could be the other way, if suppose the substrate is also negatively charged, the reverse might happen. That means the bulk concentration may be more than at the site of the reaction, and essentially that might lead to the apparent effect on the enzyme activity. Because on the left hand side I have shown the diffusional effects. You see the particle shown here, let us say a kind of a spherical particle, not perfectly sphere, but something which can be assumed to be spherical, and with a porous network, the network of pores within the particle, and the enzyme is located while on the surface as well as on the bulk of the enzyme. In fact, a very large percentage, more than 90% of the enzyme will essentially be will be distributed on the porous network within the particle. Only a very small fraction will be on the surface, and therefore now for the reaction to take place, the substrate has to diffuse from the bulk to the site of the enzyme reaction. Now here you notice it that in the case of a immobilized enzyme, unlike a soluble enzyme what we are conventionally used to, where the enzyme is in direct proximity with the substrate and can interact and reaction can take place without any resistance other than the chemical reaction, the biochemical reaction. Here, in addition to the biochemical reaction, you have another sort of limitation, that means the bulk substrate has to diffuse through the stationary film which is on the surface of the particle. Now this surface may have a thickness delta on the basis or on the basis of the properties, physical properties of the substrate molecule and the carrier particle, and now this can, of course, the stationary film essentially the thickness of the stationary film can be reduced by changing the flow properties in the reactor stream, and as we will see further that the effect of the thickness of stationary film can be reduced, and by if you just increase the linear velocity in a continuous reactor, linear feed velocity, or increase the agitation speed in a stirred vessel, in a stirred reactor. So you can reduce it, but anyway it has to transfer from the bulk phase to the stationary phase, and there might be some substrate gradient, concentration gradient in the substrate. Now from the surface the substrate has to move to the enzyme active site, and now enzyme is distributed all over the particle. Now depending on the particle thickness, let us say it is the diameter of the particle is dp, depending on the particle thickness the substrate might be able to reach only up to a part of the carrier. Let us say it reaches up to here, the substrate value comes down to 0, which will mean that a fraction of the total enzyme available in the immobilized preparation is not able to interact with the substrate, because of diffusional limitations. If the now this will depend essentially on the relative rate of diffusion vis-a-vis the rate of biochemical catalysis. Now obviously slower is the rate, the slower rate will dominate the effect. Now if suppose the biochemical reaction rate is very fast, I mean when I say very fast compared to rate of diffusion of the substrate into the carrier particle, it might happen that at a very soon after the surface of the catalyst particle the substrate concentration might drop to a very small level or even to 0 depending on the rate of diffusion is smaller than that. Or on the other hand, if the biochemical reaction rate is very slow compared to diffusion or diffusion is very fast, you might have a reasonable concentration of substrate even at the center of the particle, and therefore the apparent loss of enzyme activity may not be serious. So in case of enzyme reactions where enzymes have a very high activity, or in other words what I am trying to say, that if you are able to make a preparation which has a very high enzyme loading, that means the amount of enzyme in terms of international unit per gram of the carrier particle, if that can be increased to a significant level, then the system can be operated which is under diffusion limitations. If you are able to prepare an immobilized enzyme preparation with a very high enzyme loading, that means the catalytic rate is very high. Enzyme loading high means the catalytic rate will be very high, because after all what you are doing is the enzyme activity in terms of international units, the reaction rate per unit weight of the carrier. If that is very high compared to the rate of diffusion, then the whole system can be conceived to be totally diffusion limited. The biochemical reaction kinetics will not have a major role to play, and the whole thing will be controlled by the physical diffusion. So particularly now such a effect, now as I mentioned, now I have told you two kind of effect of diffusion. One is the external film diffusion, the other the diffusional limitation caused by the boundary layer film, inert film on the boundary as external film diffusion. On the other hand, the diffusional process within the pores of the particle are internal pore diffusion. Now the two types of diffusion, of course we will subsequently see their quantitative analysis in relation to the biochemical reaction, but what we should understand that these two processes will put you four additional resistances over and above the biochemical reaction. One is the transport of substrate from the surface to the enzyme active site, and then reverse will be transport of product. Ultimately for all practical purposes our measurements or monitoring will be in the bulk. So the product has to also transport, and it might also undergo a kind of a gradient. Now just to illustrate a little more detail on the transport behavior, you just consider a typical spherical particle with a external film shown here, now this is the substrate concentration. Ca is shown as substrate concentration at the bulk phase, that means at the surface, at the entry of the boundary layer, and now there will be a gradient of substrate in the boundary layer. Of course this gradient will depend on the thickness of the boundary layer, and you can minimize this by increasing a very high agitation speed in the stirred reactor, or a linear flow velocity, increase very high linear flow velocity in a sort of a continuous reactor. Now there is a reduction, but then in the internal porous structure the substrate concentration may undergo also a gradient. Now this gradient, it might so happen that it undergoes it, let us say it comes down to 0, the substrate concentration comes down to 0 at this point, and therefore all the enzyme present in this phase is essentially not available, and apparently it will look as if there is enzyme activity loss. So basically the enzyme activity loss as a result of diffusional limitations is a totally apparent loss. I mean if you can reduce the particle size, if you reduce the particle size to the extent that the diffusion, I mean that the rate of diffusion is enough to counter or enough to take the substrate up to the interior of the particle. That means if you can reduce the particle, let us say if this particle was only this much size, or if this particle was only of this much size, then it could happen that, and this is the center, it could happen that the enzyme might reach up to the center of the particle, the substrate might reach the center of particle, and all this particle is interacting with the substrate. So there is no activity loss, and that is what really I meant by the apparent loss of enzyme activity. That means by some physical interventions, by reducing the particle size, one can overcome the internal pore diffusion. Similarly, by increasing the agitation speed in the stirred vessel, one can reduce or totally reduce the thickness of the film, and thereby the gradient on the external film diffusion can be minimized. Now in the part B and C, you will notice the combined effect of partitioning and diffusion. You see here in the bulk phase, now the part B shows that in the carrier particle the diffusional limitations are not very high, although there is a concentration very mild concentration gradient, but the substrate is present all through the carrier particle, but the concentration of the substrate in the carrier is higher than the bulk, so there is a partitioning effect. That means the chemical nature of the carrier is has more affinity for the substrate than the bulk. In the C part, the reverse is there, that the substrate has lower affinity. That means suddenly from this boundary surface the concentration drops and there is a partitioning. So this picture shows the first one is totally diffusion. This B and C part indicate a combined effect of diffusion and partitioning. So basically these are the four major parameters or the factors which cause the various effect on the functional properties of the enzyme molecule, enzymatic reaction, and they, in fact all our effects, whenever we have to look at any particular feature of an immobilized enzyme reaction, we have to look into these four effects and try to find an answer to our observed behavior. Now let us look at some of them. The first is the effect of pH. Now very often we look, enzyme catalyzed reactions are known to have an optimum pH, and in most cases it has been noted that, particularly in the case of immobilization by ionic binding, it has been noted that the optimum pH, there is a apparent shift in the optimum pH. When you take a soluble enzyme, a native enzyme, and determine its optimum pH as against the optimum pH of an immobilized preparation, you find that the optimum pH has shifted, and now this shift is attributed mainly to the partitioning of hydrogen ions between bulk and the carrier particles. That means, as I mentioned, that the partitioning effect of hydrogen ion between the bulk and the carrier particle, depending upon the nature of charge. If suppose the carrier is negatively charged, there will be higher concentration of hydrogen ions compared to the bulk, and vice versa, and therefore in the case of a negatively charged carrier, the apparent shift in pH will be on the alkaline side, because for the higher pH in the bulk, the actual pH in the carrier particle will be lower. Hydrogen ion concentration is more in the carrier, so there will be the apparent shift in the pH optimum, because what we are considering, we are not measuring the pH in the carrier particle, we are measuring the bulk pH. So at the even higher bulk pH, at the enzyme particle, at the enzyme active site, reactive site, the pH will be lower, and so we will consider it is almost like an apparent shift in the optimum pH on the alkaline side. And the reverse is true if suppose you take a polycationic carrier, and the shift will be on the acidic side. Now the second effect of pH is on pH stability. Again the same thing, the same partitioning effect. Now particularly in some cases of the enzymes which are known to be more stable under alkaline side or acidic side, and immobilization can lead to increased or decreased stability of the enzyme depending on the their behavior. But although the pH stability as well as optimum pH in the real sense and in the real sense at the molecular level does not change, the behavior remains the same. It is only a change in the apparent change in the monitoring system, because we are measuring in the bulk and therefore the two effects are noted.
So what is how pH stability?
Now if you consider the stability effect of the enzyme with respect to pH, certain enzymes let us say are more stable under alkaline conditions. So the immobilization to a anionic polymer will be able to give you a more stable preparation with respect to pH. Consider that I am talking about stability with respect to pH, not thermal. There will be no effect on thermal as a partitioning effect, but on the pH stability, because what the pH what you are offering to the enzyme particle in the bulk is not the same at the enzyme active site. So it is reduced, and if it is let us say less stable at the alkaline pH, then even at a higher pH the enzyme particle may show a higher pH stability.
Both of them amount to one and the same.
Right, the behavior is same. The physical reason which attributes to both the effects is identical. That is partitioning of hydrogen ions between the bulk and the carrier particle. That's right. Now, as an example, if you look at some of the optimum pH of the immobilized preparation which are reported in literature, now you will notice that in most cases here the effects are mostly on ionic binding, but of course the pH effects are noted also in minor on a minor side in cases of gel entrapment and non-specific binding also, and particularly cross-linking effects also, but they may be a result of a very complex system which cannot be defined very accurately, but ionic binding can be defined very accurately. Now if you look at in trypsin, by binding onto DAE-Cephalodex, the free enzyme pH was 8.5 reduced to 7.0. Similarly on invertase, DAE-cellulose 5.4, 3. The immobilized enzyme optimum pH is reduced.
So does that imply that DAE-Cephalodex is positively?
Yes, it is in fact. Diethyl aminoethyl is that there are a lot of amino groups, so it is a positively charged matrix, and therefore the enzyme activity is reversed. On the other hand, if you look at an example here on physical adsorption onto bentonite. Now bentonite is a clay with a high negative charge, and this attributes to an increase in the optimum pH. Of course, some optimum pH changes have also been reported in the case of cross-linking, but of course their effect of change in optimum pH is a result of some other conformational interactions and not due to partitioning. I mean, in fact, particularly the last example of the immobilization of NADH dehydrogenase by cross-linking also indicates the shift of the optimum pH, but here the optimum pH shift will not be attributed to partitioning effect. It could be due to some of the conformational changes as a result of cross-linking, because conformational changes also can bring in some change in the optimum pH. A quantitative analysis of the partitioning effect can be given in terms of a factor what we can call the partitioning coefficient, the partition factor, which is the ratio of concentration of any species on the enzyme active site divided by the concentration in the bulk. And if you consider the pH effects or hydrogen ion concentration, it can be described in the two phases, that means at the solid phase in the enzyme active site and in the bulk phase by a Boltzmann type of distribution, whereas you can give the concentration in the enzyme phase, that is in the carrier matrix, given by He is equal to Hb exponential minus E psi over kT, where E is the electronic charge, psi is the electrostatic potential difference between the two phases, K is the Boltzmann constant, and T is the absolute temperature. Simplifying this, you can get delta pH as equal to 0.43 E psi upon kT.
What is psi?
Is the electrostatic potential difference between the two
What does
phases.
that mean?
That means the potential difference or the potential as a result of the charge. in the two phases. Now, if you notice again this shift, of course what I have already told, if you see the pH effect change, A is the native enzyme, B is the same enzyme chymotrypsin immobilized on ethylene malic anhydride copolymer, which is polyanionic, and the pH shift is towards positive, towards the alkaline side.
So what is H here?
Pardon?
H.
H is the hydrogen ion concentration, which you can convert into pH by changing the. Now, on the left hand side, the profile C indicates a pH shift to the acidic side when the enzyme is mobilized on polyorthenine, which is a positively charged matrix. So, basically, as a matter of fact, this has been one of the attributes in the case of immobilized enzyme preparation, where many times we require a particular operational pH for certain other conditions. For example, I must have given you an example of glucose isomerase. Now, glucose isomerase, some of the native enzymes have a pH optimum of about 7.5 to 8 alkaline. Now, obviously, an alkaline pH for isomerization of glucose is not a desirable feature, because it will undergo a lot of non-specific, non-enzymatic reactions on glucose isomerization, producing products other than fructose and it will produce sucrose and galactose and all of the sugars which are not desirable in the product. Now, a very significant property of particularly changing the enzyme apparent change in the enzyme optimum pH has been used by immobilizing those enzymes to a positively charged matrix. Now, obviously, apparently you can use the enzyme in the bulk lower pH, although the actual pH of the active site will be same 7.5 or 8, whatever is the pH optimum pH of the native enzyme, but the reaction can be carried out at a pH in the bulk which is lower, and therefore the side reactions can be totally overcome. So, that gives you another major advantage for using immobilized preparation for many of the commercial, and actually, as a matter of fact, the example of glucose isomerase is a typical example which has used such a phenomena practically. Now, the other effect of this immobilization is on the kinetic parameters. Now, kinetic parameters also undergo some apparent change, although the kinetic parameters of the enzyme does not change in real sense, but there are apparent changes in the kinetic parameters. Now, first if we consider the Km, the Michaelis constant, in most cases, as you will notice if you look into literature, that the Km value undergoes a change. Now, the changes can be of various nature, different methods have yielded increase in Km value, decrease in Km value, and sometimes no change in Km also. And now, obviously, the change in Km value essentially can be attributed to diffusional limitations as well as partitioning of substrate. Now, very often wherever the substrate is also a charged molecule, the similar partitioning effect will take place, and the substrate there will be a difference in the substrate concentration between the bulk and the enzyme active site, as we saw in the substrate profile of the immobilized enzyme particle, that in the bulk phase and in the immobilized carrier particle there will be substrate gradient, and depending on the chemical nature that will undergo, and this also Km apparent can also be related to Km of the native enzyme by the function exponential function e to the power z e psi over kt, where z is the number of charges.
So the actual Km is the same, only if you observe.
Yeah, right. It's a observed change. The Km does not change really, and the other factor
What is the number of charges on the carrier molecule?
on the substrate. It will be considered on the substrate, the charges on the substrate. Then the second reason for the apparent change in Km could be attributed to diffusional limitations. Now, diffusional limitations, the effect due to diffusional limitations on the Km have been clearly demonstrated by experimental data, because if you just use an immobilized enzyme preparation in a reactor and measure Km by either reducing the particle size you are able to come to a Km value which is of the native enzyme, which only implies that diffusional limitations are playing a role. Now you will see in some of the results now for example in one case, in one of the result reports, the Km values of immobilized glucoamylase on different substrates: soluble starch, amylopectin, amylose and maltose. Now 4 different substrates which can be acted upon by glucoamylase or amyloglucosidase. Now you see that the free enzyme, the Km values are given here on the left hand side. The immobilized enzyme, you see the Km values have significantly increased excepting on maltose where it has decreased. And basically now this also gives you a feature that the molecular weight of the substrate can also influence the partitioning effect, because the maltose being the smallest molecular weight substrate here, it can easily pass through and freely pass through and to the carrier particle, and thereby one can achieve a much higher Km value compared to the soluble one.
So please repeat the reasons for this.
So it will decrease.
Pardon?
Why should it decrease?
Km in the case of maltose has decreased.
So why should it decrease?
It decrease means higher affinity, that means the concentration of the substrate in the at the enzyme active site is much higher, much higher. So essentially what you are this concentration is essentially in the bulk, so it affects, and particularly of the higher molecular weight substrates, they behave in a reverse way, therefore their concentration usually at the site is much smaller because of the diffusional barrier, and therefore the Km value is enhanced. In fact, as a matter of fact, in most cases there is usually considered to be an increased Km value, which is not a desirable feature. This has to be taken care while preparing the enzyme, immobilized enzyme preparation, so that the Km value does not, because otherwise it will lead to a less efficient catalysis. Another data on the influence of particle size on the Michaelis constant of immobilized, the same enzyme, if you look at on maltose as a substrate, this is yes, no, this is not maltose, this is I think soluble starch as a substrate, and you are as a function of particle size, the particle size being in micrometers, the Km value increases as the particle size increases. Now this clearly demonstrates the role of diffusional limitations in the on the effect of Km, and obviously with the particle size increasing, the system becomes more and more diffusion controlled, because the particle is bigger, and say for example a roughly about 2-fold increase causes from 19.3 grams per liter to 31.6, a very significant increase in Km value. Similarly, on the external film diffusion, also the linear velocity of the immobilized glucoamylase using a 30% weight by weight 40D corn syrup. Now mind it, the data here are important in the case of starch syrup, because starch syrup when we use for hydrolysis, we use a very concentrated thick slurry, thick syrup. Even in the case of glucose isomerase, when we carry out the industrial reactions, the substrate concentration which is used is about 2.5 to 3 molar glucose, which means almost about 45 to 50% weight by volume. So it is a very thick syrup, and at such a thick syrup, the external film, the boundary film can be very thick. The film thickness can be very high, and external film diffusion in those cases where we use substrates as very thick syrup is usually very high, and external film diffusion also becomes a major parameter, and as is shown here, if you consider in a sort of a packed bed reactor with a linear flow velocity of from 8.85 centimeter per minute to almost as high as 14.5, almost 20-fold increase, gives you a decrease of 7.76 to 3, almost less than half the value of Km. That means if you are able to overcome external film diffusion, The increasing the linear velocity means you are reducing the thickness of the boundary film, and you can and that is why, as a matter of fact, when we talk of Km, we are talking only apparent change and not the real change. The other parameter which is important in the case of kinetic parameter is change in K2, that is the turnover number, or in other words you can say, if you multiply with the enzyme concentration, it becomes Vm. Now enzyme K2 change is solely attributed to conformational changes, that is, if the enzyme undergoes a conformational change, its specific activity will undergo a change. Of course, as I mentioned right in the beginning, that in some cases the change has been favorable. There have been reports, although rare, not very common, the enzyme activity increases as a consequence of immobilization. Another functional parameter is temperature, which is important for all catalyzed reactions. Now this temperature has mainly three consequences. One is the optimum temperature of reaction, just like optimum pH. We also need a optimum temperature for reaction, which is known for a native enzyme. Now in many cases there has been report in the change in the optimum temperature also. Now the change in optimum temperature now essentially is a result of temperature gradient across the bulk and the enzyme particle, and this is very prominent when you talk of gel entrapped enzyme preparations. The gel acts almost like a thermal insulating material, and therefore the temperature at the site is much relatively less as compared to the bulk temperature, and therefore the apparent optimum temperature will essentially be higher than what is required. At the active site it is the real optimum temperature, but in the bulk it is slightly higher because there is a gradient of temperature between the bulk, and of course optimum temperature has also been attributed to conformational changes, particularly in the case of adsorbed enzyme preparations or covalent coupled preparations, where there is no likelihood of any temperature gradients, or you can even notice the temperature changes in the case of a non-porous matrix, where all the enzyme is immobilized on the surface of the matrix. There is no porous network, and in those cases if there is a temperature change, optimum temperature change, it is attributed to conformational change. And of course diffusional effects are also very prominent with respect to temperature effects, mainly because the diffusion itself is a process which is very significantly affected by temperature. In fact, the diffusion process is much more sensitive to temperature effects than many of the catalytic reaction rates. The rate of diffusion can be increased, enhanced significantly as a result of increase in temperature, and therefore the diffusional effects also, and particularly in case where the system is diffusional controlled, the temperature optimum might undergo a significant change. The effect of diffusional limitation on the optimum temperature is on the conformational changes. Now conformational changes, you see, in the case of enzymes which are immobilized by carrier binding methods, for example either by adsorption or by covalent binding, where there are no likelihood of much temperature gradient in the bulk and the enzyme active site, and this can be very easily checked in case if you consider a non-porous matrix where all the enzyme is supposed to be on the surface. There is no porous network, and in those cases the change in the optimum temperature will be attributed to conformational effects only, because there is no chances of any diffusional effects, there is no chances of any temperature gradient. So all the three factors, because as you will notice that in different cases the different kind of changes in optimum temperature have been noticed. Then the second parameter which is affected by temperature, on the effect of temperature, is the energy of activation. The effect of temperature on reaction rate gives you a parameter, activation energy, and which is considered also to be a characteristic parameter for any chemical reaction. And now in the case of activation energy, again the major reason, the contributing factor is the diffusional limitations, the rate of diffusion, because as I mentioned that the diffusion process itself is highly sensitive to temperature, and the activation energy in the case of immobilized enzyme preparation is essentially a net resultant of the two rate dependent, the two temperature dependent processes, the chemical reaction and the diffusion. So, the net effect of the temperature on the overall reaction rate, which is a combined parameter between the rate of diffusion and the rate of reaction, can be different than the native enzyme where there is no diffusional limitations. Then a very important factor which is always, although in the case of activation energy and optimum temperature I mentioned that different systems have shown different kind of behavior. There is an increase in activation energy, there are reports on decrease in activation energy, similarly there are increase in optimum temperature, there are decrease in optimum temperature. Of course, there are cases where there are no changes also, but one parameter where what is understood as storage stability, which is attributed to the thermal deactivation of the enzyme with time, is always favorable in the case of immobilized enzyme preparation, mainly because of the additional intermolecular forces and intramolecular forces that are involved, which keep the process of deactivation to a minimum. In fact, in almost all cases of immobilized preparation, the storage stability has to be noted to be higher in terms of its half life. So, that is almost a proven fact, and obviously that is one advantage we have to take of immobilized enzyme for all operational purposes. Now, just as an example of optimum temperatures of various immobilized invertases, as you notice here, the native invertase has the optimum temperature of 55 degree centigrade, and by the immobilized preparation by different methods, like physical adsorption on activated carbon 40, ionic binding to DI cellulose 30, a very significant difference change, which essentially is attributed, you will notice that it is as I mentioned sometime earlier also, that the adsorption process undergoes slightly unfolding of the enzyme preparation, enzyme protein, and unfolding of enzyme protein is essentially an exothermic process, endothermic process, I am sorry, is endothermic process. So, which essentially results in a sort of a reduction in the optimum temperature. Alkylation by covalent binding 45, entrapment 50 degrees. Now, here entrapment is solely as a result of the insulation effect, the temperature gradient across the.
So the optimum then should be higher.
Which
Yes,
one?
sir.
So because there's a temperature gradient, the temperature
Right.
will be higher in the bulk than the carrier. So the optimum pH which you observe should be higher.
Right, as a matter of fact, that is right in the case of gradient, but here that means there is some conformational change also occurring. There are some minor conformational change, but not as high as alkylation or ionic binding, but there is some conformational change also occurring, which is that otherwise in fact optimum temperature should be higher in entrapment if it is solely by temperature gradient.
Sir, for better stability, so the thermal deactivation would take, like for the native one, if it was taking place at 55 degree, it was optimal. So the other enzyme, by the other processes, we should be able to increase the temperature.
No, this is not essentially, I am not talking here the stability, I am talking of the initial reaction rate as a function of temperature, not stability. Now, if you look at the data on the activation energy of various immobilized aminoacylases, I will come to operational stability in a little while. Now, activation energy for the immobilized aminoacylases, again bound by or immobilized by different methods, also undergo a change. As you will notice that both the types of behavior, there is a significant increase on DI cellulose by ionic binding, or DI Cephalodex slightly less increase, but on covalent binding there is a decrease in activation energy, and this change in activation energy is a resultant sum total of the diffusional effects and the biochemical reaction. And entrapment in polyacrylamide also results in decrease in the activation energy.
So how can we explain this entrapment? Because and the entrapping.
Why? It should decrease. The diffusion, it will entirely depend, on the preparation. You see, just by what I am trying to show that there are both kinds of effects, because the preparation can be made to be both diffusion limited as well as, if the enzyme loading is not very high, it can be biochemical reaction limited also, and the activation energy measurements will indicate that figure. As a matter of fact, the measurement of activation energy is also used as a kind of experimental data to check whether the system is diffusion limited or not. So it will entirely depend on the loading of the preparation, and not necessarily that in all cases it will be. In fact, as a matter of fact, here the figures are given under certain experimental conditions in the literature, and under which, depending on the diffusion limitation or the loading of the enzyme on the carrier, will dictate the activation energy, final activation energy parameter. Now finally, the most important parameter which we are concerned with in the case of immobilized enzyme is their use in continuous reactors or long term reactions. Whether we can use them in a stirred vessel or in a packed bed or in a fluidized bed reactor, or a variety of reactor configurations that are known to us from chemical reaction engineering, and we can see the parameter which is most important to look at the immobilized enzyme preparation. A property is the operational stability, that over a period of time when we use it, it should remain stable. Again, we measure it in terms of half life, and the various reasons which cause the loss of stability during operation are enzyme denaturation, the thermal deactivation of the enzyme, adsorption of the inhibitory substances, because the process stream, the feed stream might include certain sort of toxic substances which might inhibit the enzyme activity. Bacterial contamination is another very often faced problem in the case of enzyme reactors, particularly if you are handling, say for example, a good nutritive substrate, hydrolysis of proteins, or any other, or say even glucose syrup, it can undergo bacterial contamination. Then enzyme leakage, depending upon the method of immobilization, if you have used an adsorbed preparation, there will be a continuous reversible desorption process, and leakage might be there. And of course finally degradation of carrier itself during use, the whole matrix is under certain hydrodynamic conditions, and which might cause the degradation of carrier. The final profile, you can get different kind of profiles as far as the immobilized enzyme operational stability is concerned. Now the profile 1 you see here. Now this is essentially activity factor means E upon E naught, or rather ln E upon E naught I am referring to on this profile. It is and is a logarithmic scale. You must get a first order decay as is shown in the first case. Now in many cases these are the four different kinds of sort of operational stability that have been noted, and you will notice that they all relate to certain discrepancies in the immobilization procedure. For example, if you will notice that in the case of second profile, this one, there is an initial increase of the enzyme activity and then the normal decay. Now very often such a profile is noted in the case of immobilized cells. In the case of immobilized cells, where initial autolysis of the cell releases some enzyme, and then the enzyme remains entrapped and shows you a normal first order decay. But initial autolysis of the cell gives you some enhanced activity. The third one is there are two-step decay. Initially the enzyme is decaying at a very, its stability is very high. Suddenly there is a bacterial contamination sets in, and the decay becomes very fast, or some operational default, some fault in the operation takes place. Contamination is usually a very usual problem in continuous reactors, and therefore you have a very fast decay. In the case of fourth one, you have a very initial very high decay and then a continuous. Now this is some kind of an incomplete immobilization. For example, if you consider adsorbed preparation, some of the enzyme is weakly bound, which gets dissolved very fast, and then the really adsorbed enzyme preparation shows you normal decay. So different kind of behavior, operational activity can be seen and can be attributed to the. So now we